The broadcast is now starting. All attendees are in listen-only mode.
Good morning, everyone. Thanks very much for your time this morning. My name is Julie Petty, and today we're hosting a webinar for the goat industry. Today's webinar is focusing on a couple of different things, where we've got a presentation from Mr. Ben Swain of BCS Agribusiness, who'll be giving us an overview of a number of different information relating to the Boer Select producer demonstration site, where a number of Boer goat sires have been evaluated and their progeny assessed for a range of different traits. We've also got Dr Johann Schroder, who's the Program Manager within MLA, looking at animal health and biosecurity. We've got everyone's contact details up on the screen at the moment, as you can see, if you did have any questions following the webinar and wanted to follow up with any of our speakers today. I should let you know that today's webinar will be recorded, so if for some reason you know of someone who has not been able to dial in for the webinar, the webinar will be recorded, and the recording will be uploaded to the Meat and Livestock Australia website, where it can be accessed at a later date. All past recordings have already been uploaded to the website, so we've had 2 already, and those are up there if anyone wished to dial in and have a look at those. For today's webinar, there's a couple of ways that you can dial in and listen in to the webinar. Option 1 is to use your computer speakers, which is, I think, the default option. However, if you're having any delay or issues with feedback or anything like that, you are welcome to dial in using your phone, and the phone number and access code are there on your screen. They should also be included in a reminder email that you would have received yesterday. So today's presenters, as I mentioned, are Mr. Ben Swain from BCS Agribusiness and Dr. Johann Schroder. What we might do is begin with the first presentation from Mr. Ben Swain. You will notice that in your webinar control panel, which is on the right-hand side of your computer screen, there is an option there where you can dial in, change your audio settings, and also type in any questions that you may have for today's speakers. So I'd encourage everyone to, if you had any questions, type those in. We have got all the attendees automatically put on mute just to limit the amount of background noise and feedback and so forth coming in, disrupting the webinar. So that's the best way, if you had any questions, to get those answered. So Ben, if you're ready for your presentation, I'll change over the control panel and set you as the presenter.
No problem, Julie.
Okay.
Okay, so everyone should be seeing my my presentation now, which is genetic improvement, increasing productivity. So thanks, Julie, for inviting me along today to give a brief overview on both genetics in the goat industry, as well as a bit of information on the Boer Select buck evaluation trial that we've been running over the last couple of years. So without further ado, we'll jump straight into it. So if I can get this to work. So today we're basically going to look at 3 things. We're going to have a quick look at what estimated breeding values are about, because that's what the trial's been all about. So how do we calculate these estimated breeding values? And plenty of you are probably used to breeding values from cattle or sheep industries, or even from the goat industry. We're going to look at some key production traits which breeding values can help us with in the goat industry. And finally, we're going to have a detailed look at the results from the Boer Select buck evaluation trial using what we've learned about breeding values and production traits. So I guess the first question that everyone asks is, you know, I invest in fertiliser, I invest in fencing, I invest in staff, why should I invest in genetics? So I guess it's important, before you invest in anything else, and before you invest in feeding animals or fencing them or managing them, you've got to invest in the genes, because the genes of an animal is what sets the potential for what that animal can be achieved. You can feed the animal, feed any animal in the world the best nutrition and look after it under the best management scenarios, but if it doesn't have the genes in place to actually thrive and produce at a top level, it's never going to be a high performer. Feed and management, they provide the tools to get somewhere near the potential. So even with a poor genetic-based animal, you can still feed and manage it, and you'll have good animals. But without those genetics being in the animal from the day they're born, you're not going to get the full potential of the animal. And it costs us the same to run a poor animal as it does to run a good animal. So we might as well be running the best animals we can. The important thing about genetics and why it's a really good investment is it's cumulative. So every generation that we invest in genetics, we're getting a cumulative return, generation upon generation upon generation. And the investment is permanent. It will stay there forever. As long as we continue to maintain those genetics, it will stay there forever. It doesn't depreciate over time like a hay shed. If we continue to invest in it, it stays there forever, and it adds and adds and adds as we go. So let's talk about breeding values. So genetics are important, we know that, but what are we talking about when it comes to genetics? When we look at an animal, and we're looking there at the screen at a good-looking Boer goat, what we're looking at there is what we call the phenotype. It's what we see when we look at an animal. It's what we see when we look at people. It's what we see when we look at wheat or barley or anything. But the phenotype doesn't actually tell us how that animal performs in relation to its progeny. The phenotype's actually made up of a genotype, which is the genetics or the DNA an animal carries, and we're all familiar with those little DNA pictures that you see all around the world with the little chains of amino acids. That's the genotype. And it's also obviously influenced by the environment, which is what we see every day in the field. So how we manage an animal, what sort of a season we're having, how old the animal is, they're the environmental factors that make the animal look like what they look like. But remember, it's only the genes that are passed on to the progeny. No matter how well you feed a buck and look after him, if he doesn't have the right gene, his progeny aren't going to be any good. So it's really important when we're looking at selecting animals to bring into any sort of a production system that we focus on the genotype, not the phenotype, because the phenotype just tells us what his previous owner's done for him, how he's been managed, what season he's experienced. Simple things like if he was born as a single or a twin. If you're born as a single, you're going to have a lot better start in life than if you're born as a twin or a triplet or a quadruplet. So all of those are environmental impacts that we need to take out of the system. And that's exactly what estimated breeding values do. So estimated breeding values, or EBVs we're going to call them for short, save a bit of time, they describe only the genes. They take all of the environmental factors out. So whether they're born as a single or twin, whether they were born in a drought or a flood, whether their mother was a maiden or an adult doe, all of those issues are taken out of the EBV, and we're left with just the genes, which is what the animal, the sire or the dam, is going to pass on to the progeny. So how do we do it? How do we calculate EBVs? Well, here's a little picture that shows everything that goes into the EBV. Over there on our left, as we're looking at it, we see the animal itself. So we measure the animal for growth, for worm egg count, for carcass traits. We measure all of those things. We put that in. But on the right-hand side, we also know, in some cases, who its sire is, or maybe even who its dam is in a full pedigree slot. And we know all about those animals as well. It may have some siblings. It may have been born as a twin, so it's got a full sibling. It might have some half siblings from the same sire group. All of the information from all of those related animals go into the system. Down the bottom, we talked about environment. We know what sort of environment, whether it was born as a single or a twin, what sort of season it was when it was born. That goes into the box. Over on the right-hand side, we've got correlated traits. And by that I mean traits that are related to each other. So eye muscle depth and carcass weight, for example, are obviously related to each other. So we can get an estimated breeding value for an eye muscle depth without actually measuring eye muscle depth, because we know the correlation between growth at, say, yearling stage or 1 year of age is a correlated trait, or it relates to eye muscle depth. Heritability is the other thing. All traits are different heritability, and by that I mean the chance of that trait being passed on to the progeny. So in typical growth sort of traits, we're talking about a trait that's moderately heritable. So it's probably about 30 to 35% heritable. So the genes that an animal carries for weight, 30 or so percent of those genes are coming from its parents. So we put all of those into the box and we end up with an EBV. The more information we put into the box, the more information we've got on the animal itself, the more information on the sire and the dam and the heritability and the siblings, the more accurate the estimated breeding value is. Keep in mind that that first word in the name is estimated. If we had an infinite number of progeny, so if the sire had an infinite number of progeny, the estimated breeding value would actually be what we call the true breeding value, and it would be actually what it breeds. But of course there's no sires out there with infinite numbers of progeny, so we call them estimated breeding values. But some of these estimated breeding values have accuracies, which tells us the level of confidence we can have in them, up in the 95 and above percentages. So they're very close to true breeding values. So you can be fairly confident with a breeding value with a high accuracy that if you take that animal home, he will breed exactly what the breeding value tells us. So what are the key traits? So in goats, carcass and growth. So obviously the goat industry is an industry that's dominated by the key productivity trait being carcass weight or kilos, and it comes down to carcass and growth. So when we look at that across the various sectors of the industry, we can say, well, is that a good thing in both, say, the boar industry or the more intensively grazed operations, and is it a good thing in the rangeland selection? Well, it's obviously a good thing for both. It's certainly a good thing in the intensive industry because it's all about, like any intensively managed agriculture, it's about kilos per hectare. The quicker we can get the animal up to a weight and off the place so another one can come along and follow, the better, and same in the rangeland selection. So if we've got rangeland goats that are growing faster, reaching a heavier weight quicker, there's less animals going off the farm at no commercial value because they're growing faster, and we're getting them off faster. So it's a positive thing. People say to me, oh, but large carcass is not always positive. We don't want big goats, you know. Some of us only want small goats because we're servicing the Capredo market, for example. Or there's a grid out there that's 8 to 18 kilos. We don't want big animals. So I guess my point there is it's not about big animals, it's about getting an animal to a certain weight faster. So growth characteristics of goats doesn't say I'm going to have the biggest goats in the country, it just means they're going to grow a lot faster than goats without those growth characteristics. The other thing people say to me is that, oh, it's really tough this, you know, in an extensive environment, in the rangeland environment, how can we possibly select our animals for traits like growth? You know, we put them out there in the paddock, and the bush billies basically join everything that's out there, and if we try and put out some selected genetics, be them Boer genetics or be them, you know, just some bush billies that we select because they look better than the rest of them, it's really hard because, you know, we can't control the management, and that's a real issue. But all I can say there is that there is people out there doing that. You know, over the last 5 years that I've been involved in the goat industry and in genetics and selection, we've seen more and more people more intensively managing their goat enterprises in the rangeland environment, having some does behind wire, keeping the bush billies out, selecting the does based on some traits, selecting the bucks based on some traits. So it's not easy, but it's certainly being done, and I'd expect over the next few years we'll see more and more of that all the time. Why do this? Well, why do it? Because it makes more money, and the lamb industry has shown us exactly why it makes more money over the last 20 odd years. That little graph there, we won't go into too much detail, but it shows the increase in growth rates in lambs over the last almost 20 years. So 20 years ago we had lambs with an average post-weaning, which is the red line, of about +2. Now they're about +10. So, you know, lambs are, by that post-weaning age, which is 7 to 9 months, on average across the industry, are now 8 kilos heavier than they were 20 years ago. So, you know, it's a pretty good example of what drives productivity in the meat industry and its growth, and we can see there the lamb industry's done it. So it's a really good opportunity for the goat industry to use the learnings from the lamb industry, use the technologies that we've had from the lamb industry, and jump on board and make some of the same gains. What other traits are there? We won't go into too much detail, but obviously worm resistance. For anyone that's in a high rainfall environment, like those goats in the picture there, there's some of the trial goats down at Bethungra. And Bethungra had a terrific, that's in sort of southern New South Wales, for those that aren't aware of where Bethungra is, down near Wagga. They had a terrific autumn through winter, and the goats there were doing really well. But they were full of worm in cases. So worm resistance is a trait that we can breed for. We don't need to be drenching it every 3 weeks. Fat and eye muscle depth. Those involved in the domestic goat industry, fat and eye muscle depth is going to become critical over the next few years. It's what butchers and restaurateurs are looking for from lamb. It's what they're going to be looking for from goat as well. Those carcass characteristics. And reproduction is obviously a key one. There's plenty of evidence out there that we need to manage our reproduction in the goat industry. And that's not always to manage it in the positive direction. In some cases, it's probably to pare it back a little bit because we're getting too many kids on the ground in an intensive environment, and we can't manage them. So either way, any of these traits, we can improve, we can increase them, or we can decrease them. They're all heritable traits, and they're all traits that we can directly measure. So we can improve any of those production traits we like. So let's have a quick look at the Boer Select buck evaluation trial. There's plenty of information available on this on the web, so we're not going to go through it all, but we will just have a quick look at a few results and hopefully whet your appetite a little bit so you can go and download the full report and have a good look through yourself. So as I say, it was down near Wagga. We joined approximately 400 odd Boer does to 11 Boer bucks. They were joined through an AI program, a fairly big AI program from the goat industry, and we ended up with about 400 progeny right through to the end of the trial. We started with a few more, of course, but we ended up with about 400. And anyone that came along to the field day at Condobolin there a month or so ago would have seen those 400 progeny penned up into their sire groups. So a sire evaluation trial is pretty much it's a very common thing to do in sheep and cattle industries. We do them all the time. As far as we know, this was one of the first of its kind in the goat industry in Australia at this sort of level of detail. So the progeny that landed on the ground last September, so September 2013, we measured those for a range of traits. The traits that we've just been talking about, we weighed them every month. We did individual worm egg counts. We carcass scanned them for fat and eye muscle depth. We did a range of measured traits. We also visually assessed them. So we looked at their individually. This is every goat went through and had 8 traits measured on it: jaws, feet and legs, shoulders, back, all of the key confirmation traits that make a goat commercial or not. We've reported all of those. They all went into the KID plan system, all those measured traits, and they're reported as EBVS. So as I say, we took all the genetic, all the environmental issues out, and we can compare those animals based purely on their genes. What the trial does, and this is a really important thing, and this is what we're trying to demonstrate, is that there's significant differences in genetics in goats. We knew that would be the case because there's significant differences in genetics in sheep and cattle and pigs and chooks. But we were really surprised between these 11 bucks how much difference there was. And if there's that much difference in 11 bucks, you can imagine how much difference there is across the industry, and in particularly the rangeland industry. The difference in genetics out there in the rangeland. We did a little trial a couple of years ago, and we found that the rangeland goat is the most genetically diverse of all the domesticated animal species in Australia. So it's got a bit of everything in it. So the difference in genetics, or the genetic variability, is enormous. What that means is that we can breed for, we can select for those genetic differences, and we can make some really big gains fairly quickly. So this is a typical page out of the the results book, and for the trial, so down the left-hand side there, you can see the 11 bucks, so that's all their names and whatnot, and some of you will recognise some of the names of the studs and whatnot. The number of progeny are there, and then all of their estimated breeding values. So let's look at one in particular. So the weight. So at weaning, so W for weaning, this is the range in breeding values for those bucks' progeny, and you can see the progeny ranged from, we had 19 in one group right up to 60 in another group. So heaps of numbers of progeny, so really good, accurate breeding values. So the top buck in that trial had a positive weaning weight of plus 1.8. So you can see that there, shaded in the grey. So that meant that his weaners at that stage were 1.8 kilos heavier than the average of that of the goats in the kid plan system. At the other end of the spectrum, there's one that's got a minus 1.2 kilos. So his goats, his progeny were 1.2 kilos lower than the average. So what's the important difference there is the difference. So the difference between the highest and the lowest is 1.8 and 1.2, which is 3 kilos. So even in this little example here, the progeny between the highest performing buck for weaning weight and the lowest is 3 kilos. So you think about if you could get all your goats 3 kilos heavier at weaning than they are now, you know, just by simply using a more potent sire when it comes to weaning weight. Exactly the same goes for yearling weight. Why for yearling? Yeah, and you can see these swap around a bit. So a goat that's good for, or a buck that's good for a certain trait may not be necessarily potent in another trait, even at different ages. So in this case, you can see the guy down the bottom at 4.8 was the trait leader for yearling weight. So he's 4.8 kilos heavier than the average. And the bottom one, I think, is the one right at the bottom, which is minus 0.5. So again, you know, over 5 kilos difference between the top and the bottom. So yearling at 12 months of age, when a lot of people are turning off goats, if you had them 5 kilos heavier across the board just by using the right sire, it makes a big difference to the bottom line. Same goes for all the other traits. So eye muscle depth is the same calculation. So you can see there we had a trait leader at 1.1 millimetres. For those interested in domestic goat marketing, yeah, 1.1 millimetres of extra eye muscle depth is pretty important versus guys that are minus 0.3 millimetres. So the one that's a bit opposite is the WEC. The WEC, we're looking for negative traits. So keep in mind, we're not always looking for positive things. So worm egg count, you want less worms in a goat than more worms. So in this case, there's a buck there that's had his progeny average minus 30% for worm egg count. Yeah, so he had his progeny had 30% less worms in them than the average, plus versus one there with plus 45. So a huge difference in susceptibility to worms. So for anyone in the intensive industry that's breeding animals in a higher rainfall environment and susceptible to worms, or even in the rangeland environment, you get wet summers, you know, worm egg counts, I know they're a distant memory, but worm egg counts are you know, pretty important, and breeding for worm resistance is a key thing that the sheep industry has really thrived on over the last few years, and the goat industry would benefit from looking at as well. There's a couple of indexes there as well. So what indexes do is combine all of those traits in a couple of different production systems and sort of give you a summary of what each goat does. So just having a quick look at the indexes, we talk about 2 indexes in the trial. A carcass plus index, which is the red line, which is really when you breed using goats as terminal sires. So if you were using say a Boer genetics in a rangeland environment to increase growth rates, you'd be looking at the carcass plus index. If you were looking at for you know, Boer genetics in a Boer environment or in self-replacing herds, you'd be looking at the SRC, which is the self-replacing index. So a couple of different indexes there, and depending on what you're breeding animals for. I did mention we looked at visual traits, and we won't go through all of them because I'm chewing up too much time already, I think. So we did measure, though, I'd say 8 different conformational traits. So here you can see the jaw, legs and feet, shoulders, back and hips and horns. And really these are expressed as deviations. So in these things we're looking for negative results. So we're looking for animals with less faults. So in all of these traits, 1 is good. So a jaw 1 is a good jaw, no fault. A jaw 5 is a heavily undershot or overshot jaw, and 2, 3 and 4 are somewhere in the middle. Legs are the same. 1 score legs are good straight square legs and good hocks and pasterns, up to 5 being severely hocky or rolled in the toes or turned out in the feet. So we're looking for animals that have more score 1s than anything else. So you can see there that we highlight in grey which animals are the ones that have the best conformation. Particularly important, if you're taking these sort of genetics out in the rangeland environment, they've got to be able to walk, they've got to be able to travel, they've got to be able to compete with the bush billies, so you can't tolerate any faults in the rules. And that's how we did the visual trait. So we put every goat, all these 400 in a box like that, and we had a independent class each and every individual goat. It was a pretty interesting day. We also did reproduction, so we looked at testicles, we looked at udders and teeth, because udders and teeth, particularly in boars, are said to be a bit of an issue. So we were keen to look at those. We also, for boars, we looked at colour pigmentation, which is obviously key for the boar breed, maybe for other traits, not other breeds, not as important, but we looked at all the same. And all of those information is in the report. So I think I've taken up my 20 minutes, so I'm just going to leave you with that useful links page. So there's lots of information about goat genetics and tips on how to use it. And down the bottom there you can see where you can get that information from the Going Into Goats Guide, which is sort of a great summary of all things goat selection and goat genetics. So hopefully I've given you a little bit of food for thought, and I'll pass back over to you, Julie.
Thanks very much, Ben. Look, just a quick reminder to everyone who's dialed in, if you did have any questions for any of our presenters today, you can go down to the questions tab in your webinar control panel, which should be on your right-hand side of your screen. You are able to type in whatever your question is there, and we can read that out and ask the presenters. So, Ben, I just had a couple of quick questions. If we go back to that first table you showed in your presentation.
That one there?
Yes. So looking at that, if I was looking at all of these different size and I was looking at one I would be interested in using on my property, would obviously, as you were saying, depending on the trait, the goat with the highest or the lowest, I should say, worm egg count may not have some of the other traits that I've been looking for. So would I be best to look at these indexes to get a bit of a, I suppose, to average out the different results?
Yeah, it certainly, and that's exactly what the index is for. The indexes are designed as almost a drafting gate to say, look, I'm only interested in the top animals here. Let's have a look at the index, which will take out, on average, the top goats as we draft them out, and then we can look at the individual traits. But it's really important that no matter how good the indexes are and how closely related the indexes are to your own production system, you've got to look at the individual traits. And there's a really good example in that trial in that we've got this buck here. It's -30 for WEC. Like worms don't worry him at all. He's very resistant to worms, but he's not growing much either. Look at his kilos over here, and that's reflected in the index. But his weight traits are really below average. So you've got to get that balance. Sure, he's not going to have many worms, but he's also going to produce fairly small goats. So we're sort of looking at something, probably a bit more balanced. Whereas at the other end of the spectrum, this fella up here with a plus 45% WEC, he's full of worms, but he actually does produce some heavy goats. But do we want really heavy goats if they're going to be full of worms? So it's really, you've got to look at those individual traits, and you've got to base your decision on the traits that are interest to you. If you're not interested in worm egg count because you're not in an environment that worms are a problem and you don't mind drenching them once a year, this bloke here is a standout buck because he's got, the third buck down because he's got such good growth traits. If worms don't worry, he's the man for you.
Yeah. Thank you, Ben. Very difficult decision looking at those. So if anyone is interested, what we'll be doing after the webinar is circulating the presentations along with some of the links to where you can find additional information and copies of the report that Ben has been speaking about in his presentation today. I just had a question from one of the attendees where we're looking at those worm egg counts and so forth, and the question is around, you know, does this mean that we're not understanding how worm resistance and so forth works? And I think partly that's going to be covered off through our next presenter, but Johann or Ben, did you have any comments initially around that? I presume that's talking about the worm egg count readings there on the table.
Yeah, I think there's two issues, I guess, from a genetic, or one's a genetics point of view and one's the management point of view, and that's what we're about to talk about with Johann. But from a genetics point of view, you know, we really do need to be focusing on worm egg counts because, you know, resistance to all sorts of compounds of chemicals are becoming fairly common, and you know, genetics is that cumulative and permanent way that we can fight worms. But I'll leave the other side of it to Johann.
Okay.
Thanks, Ben and Julie. Am I live, Julie?
Yes, you are.
Can you hear me? Okay.
Yes.
When one looks at the host-parasite relationship, you're basically faced with a choice between selecting either for worm resistance or worm resilience, and the two are not quite the same. In the case of worm resistance, you're selecting for an animal that will carry fewer worms and will have a lower worm egg count. If you select for resilience, you're selecting for an animal that, despite the fact that it might be carrying a large number of parasites, don't seem to suffer an ill effect. So I would say that buck number, third one from the top, is one of these resilient animals. Despite the fact that it carries a lot of worms, it's able to perform well. And the one at the bottom, you know, with the lowest worm egg counts is a resistant animal, but maybe not as productive. And basically one has to decide which one of those you want to go for. The one downside I see if you were to go for the resilient animal, in other words, the one that produces well despite the worm burden, is that it will also be a grave contaminator of your pasture. So it'll be excreting a whole lot of eggs onto the pasture, which then pose a challenge for other animals that might not be as resilient as he is or his offspring are. So it's not an easy decision to make, and one has to tailor it, I think, to the individual circumstances of the enterprise.
Thank you very much. We might just have one last question for Ben. What's the difference, or can you elaborate on the difference between the two different indexes on the table?
Okay, so the self-replacing index, the FRC index, is an index which is designed for people that are breeding their own replacement does. So it has a large reproduction component in it, and it's got things like maternal weaning weight as a component, and number of lambs weaned, and WEC, for example. So those things that we want to breed for in a permanent nature. So if we're going to continue to retain animals that we breed and breed out of them, so in successive generations we're compounding those genetics, that's the index we'll be using. The Carcass Plus index is really the index that you use if you're going up there to buy a buck, and all of these progeny are going to be terminal progeny. So they're all going to be slaughtered at some point, and you're not going to be breeding out of them. So, you know, the fact that if it's got good maternal weaning weight, which is how many kilos a doe can wean each year, that's not really important if that doe's never going to raise a kid, because she's going to end up on someone's table. So the Carcass Plus index is all about weight, eye muscle depth, and fat. So they're for those producers that are just looking for heavier carcasses, better carcasses, and aren't going to breed out of those animals. So they're at either end of the spectrum, and you know, they won't both suit a producer. A producer would be either using one or the other, in most cases.
Great. Thank you very much, Ben. That was an excellent summary there. Look, what we might do, Johann, if you're happy, then we might move on to your presentation.
Yep, that's good.
So No worries. I will just bring up your presentation on the screen and we'll kick on with that. Okay. All right. So, sorry about the delay there. Okay, Johann, are you happy to kick off now? I'll just give a quick introduction. Johann is our Program Manager within Meat and Livestock Australia for the Health and Biosecurity section. He's a veterinary parasitologist and is going to provide an update on the issues associated with and control options for internal parasites, as well as give you a bit of an overview of some of the supporting resources and documentation that producers can access, mostly for free online. So, Johann, the screen's up and on the screen, and we're ready to kick off.
Thanks, Julie. I can't see the first slide, so I don't know what I need to do.
Oh, hang on. Sorry, I've forgotten to share my screen. There we go. Okay.
First up, I think an apology is due. There's no way in the world can this short presentation tell you all that you need to know about parasite management. It's going to be a fairly general overview, so you can launch straight into the next one. Thanks, Julie. Just in general terms, what I would like to say is that we must not forget that parasites, it's actually quite simple. Parasites are a hygiene problem. Before man came along and domesticated animals, plant-eating animals were free to roam. They practiced almost a system of natural pasture spelling, which of course is self-cleansing because parasites don't live forever. It also meant that stocking rates was sustainable. The moment a patch of grass or forage was exhausted, the animals moved on in herds and flocks. And there was also, parasites contributed to this process of natural selection. The weakest animals were often the ones that also carried the most parasites. They lagged behind and they fell prey to the predators that followed them. And then, of course, man came along and introduced a whole set of things that changed all of that. We put in fences and introduced a system of set stocking and rotational grazing that we controlled instead of letting Mother Nature look after itself. We were forced by population growth to increase our demand of these animals, so we increased stocking rates, and it was all driven by the quest, and it still is driven by the quest for greater production. And we introduced movement control. We stopped these animals from roaming as they did before, and so we increased the amount of dung that they deposit on the pasture where they actually live and eat and exist. And of course, if we're talking about goats and the effect that all this has had, is that they've been deprived of their natural tendency to browse rather than to graze. I just want to add a little bit of an aside here. It's interesting to me that people who don't like goats, and goats often get a bad rap, like to show pictures of goats in desert areas of North Africa and the Middle East, and you don't see anything other than goats, and then the logical conclusion these people think they jump to is that it must be goats that caused the overgrazing and caused the desertification of that environment. And my counter-argument is no, they are there because they're the only animals that can still survive under those conditions. They are extremely hardy and resourceful little animals. Next slide, thanks, Julie. So for me, there are a few basic rules of parasite management, and the first one is that you avoid contact between host and parasite, and one of the ways of doing that is a system of pasture spelling and pasture rotation. As I said at the earlier slide, parasites don't live forever. They also die of old age like we do. They're also exquisitely susceptible to many environmental conditions, such as drying out, such as ultraviolet radiation. If it's too hot or too cold, they're not going to last. So if we can leave a pasture to lie for long enough, most parasites will eventually die. Of course, that doesn't gel very well with modern demands for high productivity, and hence you need to strike a balance somewhere. You can also, goats are very well equipped to avoid contact with internal parasites, because if they're given the chance, they would prefer to browse rather than graze. And of course, first of all, worm larvae don't climb trees, which goats do. And then secondly, browse contains a lot more plant tannins, which have been shown to have an anthelmintic effect. It's also true that if you, try and feed animals plants that contain too much by the way of tannin, it impacts the palatability of the plant material, and it also impacts digestibility. But there's no doubt that tannins have been shown to have an antiparasitic effect. Another way, which might not be practical under all circumstances, but which has been shown to work in others, is to introduce a system of cut and carry, where the pasture is actually mown and taken to the animals and fed in hay racks, and the animals are actually kept indoors and in this way separated from their droppings, and they are fed clean feed which has not been contaminated with dung and hence with worm eggs and worm larvae. Another basic principle or basic rule from your parasite management is to avoid the introduction of new parasites. If you have an enterprise and you're fairly on top of your parasite management problem, be careful not to introduce new, possibly drench-resistant strains onto that property. So if you have to buy in animals, put them in quarantine first of all. Give them a quarantine drench. Make sure, or do a worm egg count on them to make sure that they're not bringing in new parasites that might not be susceptible to the drenches that we have available. And then, if all else fails and you can't manage parasites by following these basic rules of separating host and parasite, then by all means use a chemical, but make sure that it's the right one. And in the next slide, Julie, if you look at the going into goats guide, this table that I've got there basically comes out of the gig guide and basically just gives names to the different internal parasites that we get, and there are roundworms, nematodes that occur in the stomach and the intestines. The two types of lungworm are also nematodes, and then of course there are flatworms like the liver fluke, which parasitizes the liver, and don't forget that liver fluke have an indirect life cycle. In other words, there is an intermediate host in the form of a freshwater snail. So if you have a potential snail habitat on your property, it's relatively simple to control liver fluke in that area simply by denying grazing livestock access to those fluky habitats. And then, of course, coccidia is a very important internal parasite. It's not a worm. It's a microscopic little protozoan parasite, but can cause devastating diarrhea, enteritis in young animals. Luckily, this is a parasite which is quite susceptible to the host's immune system. So if animals suffer from coccidiosis, it's usually the young ones. Next slide. Thanks, Jules. So if we're going to use chemicals, which chemicals do we use? And in the top left-hand corner there, I've got the URL or the web address for the Australian Pesticide and Veterinary Medicines Authority. These are the people that register chemicals for use in agriculture in Australia. And if you go to that website and you click on that link to the PubCRIS, which is the public database for registered chemicals, and you use the word goat, this is the top red circle, as a search term, you will get 147 hits. You can then export these. There's in the top right-hand corner, there's a grayed out block that says export CSV. You can export these search results in the form of a spreadsheet. And if you then filter in the product type column, this is the third column from the left in the bottom table, and you filter on parasiticides only, you'll see that there are 38 remedies registered for goats for use against parasites. So there is a large number of chemicals that one can legitimately use which have been registered for goats. In the next slide, I go into a little bit of more detail why I bang on about the use of registered chemicals. And first and foremost, chemicals are registered by the APVMA for the protection of consumers. The most important bit of information that the APVMA looks for in registering chemicals is the safety data. And safety in terms of the toxicity. How toxic is the chemical that is used in the remedy that you are using? And remember that there are different levels of toxicity, whether a single dose is going to be harmful, in other words, the acute toxicity, or whether one can tolerate, or the animal can tolerate, a long duration of smaller doses, the so-called chronic toxicity. It's also important to know if the chemical might be responsible for causing birth defects. I mean, we need to, all of us still know the word thalidomide and the horrors that result from chemicals that have not been properly tested before. And then, of course, there are chemicals, we know that cause cancer. We want to make sure that if we're going to use a chemical in a food-producing animal, it doesn't do that sort of thing. So consumer safety is top of mind when the APVMA registers chemicals. Secondly, it's also a consumer protection measure to ensure that the chemicals that they register are actually effective. So, and that they are effective under our local Australian conditions and in different environments. So the APVMA, for instance, requires that remedies are tested in the north and in the south of the country to make sure that they work under different climatic conditions. There's a further consumer protection measure, and that's to ensure the actual quality of the product that you buy off the shelf at the reseller. The APVMA specifies the quality of the raw materials that go into the product, the standard of packaging, and the quality of the finished product. So one can, nobody's going to stop you walking into a reseller shop and buying a remedy off the shelf and giving it to your animals. You're on your own recognizance. But if that remedy is not registered for that purpose, you run a number of risks. If it turns out the remedy doesn't work, there's nobody you can complain to. One of the conditions of registration from the APVMA is that if a remedy doesn't work, if a chemical doesn't work, the registrant, the company that has applied for registration for that remedy, is obliged to investigate that problem and to come up with an answer or some form of compensation for the user. If you use that remedy off-label, you do not have that recourse. Same in the case of a safety problem. If you've used the remedy and it's caused some or other harm to the animals, you don't have a claim on that company whatsoever because the remedy is not registered for that purpose. And then I think the thing that makes the Australian livestock industry more nervous than anything else is the possibility that if you've used an off-label remedy and you've not observed a withholding period, and if the remedy is not registered, we don't know what the withholding period is for that drug, you run the risk of either have violative residues in the meat that can be harmful to consumers, to human consumers, or even more important from a financial point of view for the Australian economy is that you run the risk of those remedies being picked up in an overseas market and our export ability being curtailed. So those are very real risks of using remedies for a purpose that they're not registered for. Next slide, thanks, Julie. So, yeah, as I said in 2 slides ago, there are 38 remedies that are registered for parasites in goats. In the case of anthelmintics or worm remedies, 4 different chemical classes are registered. Now, the problem we face is that there is resistance in nematodes, in gastrointestinal roundworms, to all 4 those chemicals in sheep and goats today. And so it's a problem. And so one is faced with the option of either using some of those registered remedies in combination to overcome resistance, but other than that, there's not a heck of a lot one can do. There are 2 new drenches on the market, and they are listed there. And it's a sad fact that, the top one of the list, which was first on the market 4 years ago, is already blighted by resistance in a number of roundworm parasites, both in sheep and in goats. So that remedy is not even registered for goats, but goat producers have been using it off-label, and 4 years down the track, it has a problem with resistance. And then the latest product has just occurred on the market fairly recently. It's a combination of 2 active ingredients, but I think I'm pretty safe in predicting that as sure as the sun will rise tomorrow, it won't be too long, and there will be resistance problems with this new remedy as well. So chemicals have to be used sparingly and wisely. An alternative might be to use a vaccine, and there is hope on the horizon. People listening to this webinar will no doubt know that a product called Biosecurity was launched in Armidale about a month and a half ago. It is a barber's poleworm remedy. It is registered initially for use in suckling lambs. Work is continuing to extend that efficacy claim to include both yearling lambs and adult sheep, and also, as we speak, work is underway to generate a claim so that this vaccine can be used in goats as well. We know that it works. It's certainly effective in reducing the number of worm eggs that are specifically barber's pole worm eggs that are excreted onto the pasture. So over time, with the vaccine being used regularly, it can lead to a sort of a cleansing of pasture and a great reduction in the number of infective larvae that can endanger stock grazing on that area. That picture of the goat buck nibbling on a branch I've chose that on purpose. I admire these animals greatly for their ability to browse and to survive where many other animals will not. Next picture, Julie. I'm not sure what comes on the next slide. Let's have a look. I think it's a sort of a, oh, okay. Similarly to what Ben did, there is a list of MLA, and as Julie said, free of charge resources that you can access via the website. First of all, there's the Going Into Goat guide, and the module 9 is the one that concentrates on parasites. Then there are 2 research reports which you might find helpful. The top one, number 14, lists a whole lot of chemicals that can be used both for internal and external parasites. And the second one, number 89, is one where we actually went and looked at the remedies that are registered and made sure that the dosages, the withholding periods, and the export slaughter intervals that are listed on their labels are actually supported by experimental data. And then there's a bit more information on the Biosecurity claim, which is under development. And then another activity that MLA is engaged in the interest of the goat industry is engagement in a current initiative of the Department of Agriculture to try and improve access to registered chemicals for small industries. Now, there's been great interest in the past, and people have been clamoring for the issue of minor use permits of remedies registered for example, sheep and cattle to be used in goats. And of course, it's not quite that simple. And people who make that claim then quote the example of large numbers of minor use permits that are available in the horticulture industry. And the quick and easy answer is that in that case, the remedy is already registered for a particular crop, and all that gets added in the case of a minor use permit is an additional pest. If we're looking at the livestock industry, it's not that simple, because goats are not just sheep of a different color or small cattle. They are a totally different species of animal, and hence one would have to generate residue and toxicity data for these animals. And so one of the activities that this Department of Agriculture program is engaging in is to try and see whether Australian registrants can't gain easier access to data which already exist overseas, so that we can try and avoid duplication as far as possible. I think the last slide is the next one, Julie, where I just give a bit of a recap and a take-home message. Don't forget, parasites are a hygiene problem. Humans these days very seldom need to be treated for parasites because we practice good hygiene. And the more intensively one keeps animals, the bigger the problem becomes, because the animals no longer have a chance to rotate to cleaner pasture, and they are forced into closer contact with infected animals. So where do these worms come from? In the case of internal parasites, it's usually a contaminated environment. If it's roundworms, the worm larvae hatch out of eggs that were excreted in feces or in dung, and the worm larvae sit on the plant material that those animals are forced to eat. Coccidiosis have no means of moving by themselves, but if you confine animals to pens, I think the most common source of infection would be use of dams that lie down in contaminated pens. Their udders and their teeth become contaminated with these coccidiosis, and the suckling young lambs or kids pick up the infection that way. In the case of lice and mites, which are obligatory external parasites, it's close confinement of animals that are infected with those that are clean that cause a transfer. And other external parasites like ticks, fleas, and ticks and lice and flies, and they tend to be more. There's a larger free living portion to that life cycle, and so it's an environmental contamination. If all else have failed and you cannot avoid having to use chemicals, just make sure that you use the right chemical for the particular parasite. Make sure that you use a chemical which is effective on your property. In other words, check the resistance status of your parasite population. Use the right dosage or the right dilution if you're using something for external parasites, and make sure that your application method is correct. And in other words, so that the chemical will actually work. Otherwise, it's just going to be a waste of money. And I think that's the end for me. Thanks, Julie.
No worries. Thank you very much for that presentation, Johann. That was very enlightening. Just a reminder, everyone, if you have any questions, please just type those into the questions panel within the webinar control options there. Just a couple of quick questions, Johann. You mentioned that the project that's underway for BioSecurity is due to wrap up next year. Is there any indication when the product will be available and on the market for goat producers?
I'll put on my rose-colored spectacles, which is basically almost a permanent fixture for me, Julie, and say that everyone is hopeful that we'll have that claim, or the producers will have that claim on the label by the next summer season, so late spring of next year. That is predicated by the activities of a lot of people, unfortunately, over which we have little control. One is that the actual experiments will be completed in autumn of next year, so we're looking March, April-ish. Those data will then be written in a report form and submitted to the APVMA. Hopefully they'll be able to evaluate the data and give the yay or nay for that claim to be added to the label within the next few months. And of course, once they've done that, the new labels will have to be printed and so on. So if all goes well and everything falls into place, by the time the next summer in the latter half of next year comes around, BioSecurity will be registered for all ages of sheep and for goats as well.
Okay, excellent. And there's currently some trials underway for that program. Is that based in a particular area or with a couple of different producers? Yes.
There are two aspects to the goat work which is currently running, and one is to actually prove that the vaccine doesn't cause undue problems when used in goats. Not that we expect some, but it's always nice to have the numbers under one's belt. So there's a trial where a large number of goats are being injected with the vaccine just to prove that there are no untoward vaccine reactions. And then there are efficacy trials being done in goats on 3 or 4 different properties where the goats are exposed to natural infection and they are then vaccinated and their worm egg counts are followed. This work is being done mostly in the New England because that's the Barber's poleworm capital of Australia. And just a word of caution, it's a little bit disappointing that the vaccine is only effective against Barber's poleworm, and the researchers have reassured us that they are looking at developing a vaccine or developing this vaccine further so that hopefully in future it will also contain antigens for the scour worms. But at this stage it is purely a summer rainfall area Barber's poleworm vaccine.
Thank you. We just had a question coming through. Is there similar information on the APVMA website for cattle and sheep regarding chemicals, etc., to use on specific parasites?
Absolutely. So if one looks at that slide that I showed, and you enter either sheep or goats as your search term for the host, instead of so in that top box, you will get exactly the same information for sheep or for goats. I find it easier once I've done that to export that table into an Excel spreadsheet, because then you can play around with it, because in some instances the APVMA database might be a little confused, and you need to sift through, because there's sometimes things slip through the cracks, and you get anomalous results with that search term. It can happen that you get a herbicide or a fungicide in there that clearly doesn't belong. So I like to look at them in a spreadsheet format.
Okay, and when you're doing fecal egg count, how do you know what to look for when you're doing those? Is there a course that people might be able to go on to get a bit more training on that, or kits available?
The answer to both those questions is yes. There is certainly a lot of information available on how to do this, but I have to. Although doing a fecal worm egg count is a very crude technique, it's been around for decades. It still requires a fair degree of experience and expertise to get consistently good results. You need to weigh out the amount of dung that gets used, for example, very carefully. You need to have the right equipment. The little counting chambers which are used are specialized bits of medical equipment. I mean, anybody can buy these things. You need to do it under a microscope, and anybody can buy a microscope. But it is necessary. I would urge people who want to do this themselves to make sure that they get the proper training, and maybe a good place to start would be to go to one of the diagnostic laboratories. Either diagnostic laboratories belonging to the different state departments of primary industries, or even private diagnostic service providers like Vet Health Research in Armidale.
Thank you. And this final question is perhaps better answered by you, Ben. How predictable is worm resistance in breeding? I know we mentioned that a little bit in your presentation.
Yeah, so worm resistance is a lowly heritable trait. It sits somewhere between reproduction, like number of lambs weaned, and carcass traits, which is moderately heritable. So it's not as easy to breed for worm resistance as it is some other traits. So what that means from a breeding point of view is that you need more data to get a similar accuracy breeding value. So that's where those accuracies are really important to look at. So if you've got a sire with 20 progeny in the system, the breeding value for worm egg count might only be 50 or 60% accurate, versus one for growth would be sort of 80 or 90%. But the more data and the more progeny and the more animals that are recorded for worm egg count, the higher the accuracy for the breeding value, and the more predictable that breeding of that animal will become.
Excellent, thank you, Ben. And we're just running out of time, so we might just go with one final question. And this is around, I suppose, more of the diagnosis. How would producers know what is affecting their herd or flock, so that they can implement improved herd management practices?
Wow, that's a big question, and I guess there are a number of clinical signs of disease, and unfortunately very few of them are specific. The worms, for instance, Barber spellworm sucks blood, so one of the obvious first things that it does is to cause anemia, and you can assess anemia by looking at the animal's mucous membrane. So you can either evert the eyelid and look at the inside of the eyelid to see whether it's a nice healthy pink color or whether it's pale, and if it's pale, it's a fairly easy conclusion that the animal is anemic. Now, Barber spellworm sucking blood is not the only potential cause of anemia, but it's a pretty important one. If, on the other hand, the animals are exposed to scour worms like a small brown stomach worm or black scour worm, they will be scouring because they'll have an enteritis and they'll have loose stools, and so their backsides might be soiled, or they'll you'll see that they've got diarrhea. The next clinical sign, I guess, that one will see is that the animals will lose condition. In the case of again, if we look at Boer spillworm, it's a very acute condition, and animals will die before they become thin. In the case of scaleworms, they will lose condition and they will fall behind. So I guess good stock people know what their animals look like when they're healthy, and they know what their animals look like when they're sick. And animals that are sick, regardless of what type of disease it is, they can lose condition, they can become pale, they become listless, they lose their appetite. So those are all clinical signs of disease, and one has to examine such animals carefully in order to be able to make a decent diagnosis. And it might be necessary, if there are a number of animals that are affected and which show the similar clinical signs, it might be worthwhile to sacrifice one of them and actually do a necropsy and see what lesions one can actually see in the carcass.
Okay. Well, thank you very much for that. Look, we're just running out of time, so we might end the questions there. I'd like to say thank you very much to everyone for making the time to dial in for today's webinar, and thank you in particular to both Ben and Johann for your time in making the presentation for us today. What you should be seeing up on your screen at the moment is contact information for myself, Ben and Johann. If you had any other questions or comments following on from today's presentations, please do squirt us an email. I should also mention that if you did have any questions or wanted more information about either looking at the previous recordings for the other webinars or finding the full report that Ben has produced in relation to the Boer Select buck evaluation program, those are both available from the MLA website on the Going Into Goats page. And as I mentioned earlier, what we'll do following this webinar is circulate some information and links where you can go to if you did have any other questions or wanted to find out more about using KidPlan, for example, or parasite management and so forth. So thank you once again, everyone, for your time. Today's webinar has been funded through the goat R&D levies through Meat and Livestock Australia, and we do have another webinar coming up again in the new year, which is looking at grazing pressure and a case study involving wild dog control out in regional Queensland. So look, I should also mention that if anyone has any further comments or ideas or suggestions for future webinars, please do get in touch with me. We'd be happy to get any further suggestions of speakers or topics that producers would like to hear about. And without further ado, I'd just like to say thank you once again to everyone, and we'll hopefully catch up with you at the next webinar. Thanks again for your time. Goodbye.